Hello friends, welcome to Creative Medicine. In this lecture, we will learn about.
Hello friends, let us now learn some important points about the chemistry of nucleic acid. So if you see, nucleic acid is equal to a polymer of nucleotide, which is a mononucleotide which is linked with other nucleotide by a bond which is phosphodiester bond. So what are these nucleotides? These nucleotides have a nitrogenous base which is linked to a sugar which is pentose sugar like ribose or deoxyribose, and this is also linked to a phosphate group. Okay, this together we call it as a nucleotide. The main properties of nucleotide are these are polyfunctional acids. They are actually negatively charged. Okay, and these nucleic acids will absorb UV light of wavelength 260 nanometers at pH of 7. These will absorb UV light at wavelength of 260 nanometers at pH of 7. Then once these okay, this is about the chemistry. So if you see, we will see the nitrogenous bases. The nitrogenous bases can be divided into two types. We have purines and then we have pyrimidines. Okay, so first purines are again of two types. We have adenines and we have guanine. Now in the adenine, if you see, I will just draw the picture of adenine and guanine. So this is the nitrogen atom. 1 minute. Right, so this is the purine ring. So this is a purine, and to this at 6th position we will add amino group. So this is 6 amino purine. So adenine is a 6 amino purine. Then if you see guanine, if you see, I will draw the same first, the purine ring I will draw the same thing. Right, so this is the same thing. Okay, but here oxygen is added. So this is, and also there is amino group at 2nd carbon. So this is mainly 2 amino 6 oxopurine is guanine. Guanine is 2 amino 6 oxopurine is guanine. Now what about the pyrimidines? I will draw pyrimidines in the next page. Pyrimidines are divided into 3 types. We have 3 types of pyrimidines. One, we have thiamine. So in the thiamine, if you see, first we have a pyrimidine molecule. Okay, this is nitrogen, right? To this pyrimidine molecule, if you add a oxygen bond at oxygen at 2nd and 4th position, and this is 2nd position, this is 4th position, we are adding oxygen group and we are adding methyl group at 5th position, then we call it as thiamine. That is 2,4-dioxo 5-methyl pyrimidine is the thiamine. 2,4-dioxo 5-methyl pyrimidine is thiamine. Then we have cytosine. So in this cytosine, first I will draw the same picture. Okay, right, here at 2nd position there is oxygen group and at 4th position there is amino group. Okay, so this is cytosine, which is called as 2-oxo. Okay, 2-oxo 4-amino pyrimidine. This is 2-oxo 4-amino pyrimidine, which is cytosine. So if you see, this is 2, 3, 4, 5. Similarly, this is 2, 3, 4. So it is 2-oxo 4-amino pyrimidine. Then we have uracil. So if you see, in the uracil we have, again I will draw the same pyrimidine group. So this is the pyrimidine group, right? Right. So to this pyrimidine group, at second position and at fourth position, you see 2 oxygen group. That's it. Okay, so here you have 2,4-dioxopyrimidine. Okay, so this is the different types of pyrimidines and different types of purines. Okay, now, so yeah, these are the main purine bases. Are there any additional ones? Yes, we have minor purine bases. So the examples of minor purine bases are, we have one hypoxanthine. Hypoxanthine is 6-oxopurine is hypoxanthine. Then we have xanthine, which is 2,6-dioxopurine is xanthine. Then we have uric acid, which is 2,6,8-trioxopurine is called as uric acid. So these are minor purine bases. Then we have learned about the normal nitrogenous bases. Very good. But along with these nitrogenous bases, we also have something called as modified nitrogenous bases. These modified nitrogenous bases will be dihydrouracil. These include dihydrouracil. Then we have pseudouracil, right? Then we have 5-methyl cytosine, which is used as modified nitrogen base. Then we have dimethyl amino adenine is also used as modified nitrogen base. And we have 7-methyl guanine. So these are the compounds which are used as modified nitrogen base. Okay, so this is about the nitrogenous bases. Then we will see structure of pentose sugar. Okay, now if you see the pentose sugar can be divided into 2 types. So we have one which is 2-deoxyribose sugar, which is present in DNA. Then we have D-ribose sugar, which is present in RNA. So I will draw the structure of both of them. So this is a ribose sugar. So you have something like this. So here if you see, there is at second position, there is no oxygen group. Here you are not seeing any oxygen group. That is the reason why we call it as 2-deoxyribose. Okay, I will draw ribose so that you get a clear idea. Okay, so if you see, just everything is similar in both these sugars, but only in the second position. Here you will see a presence of OH group, but here you will see absence of oxygen. So that is the reason why we call it as 2-deoxy, because this is 1, this is 2. So 2-deoxyribose is DNA, whereas deoxyribose is present in RNA. Okay, now to this bases, where are phosphate group attached? Here this H plus is removed and here phosphate group is attached during the DNA formation. Even here if you see, this OH is removed and here there is phosphate bond formation. And then if you see here, you will see the nitrogenous base being attached. In this ribose sugar, you see here nitrogenous base being attached. This complete structure we call it as the DNA nucleotide. It is called as nucleotide. Group of these nucleotides will form DNA. Okay, now right, so this is about the nucleotide. Then there is one more word which is important, is about nucleoside. We have something called as Nucleoside. See, simple. I will just tell you what is nucleoside. Nucleoside is if there is nitrogenous base and if there is pentose sugar and if there is no phosphate group, then these two together we call it as nucleoside. Okay, so there is linkage of first carbon atom of ribose or deoxyribose with first N1 of pyrimidine or N2 of purine. That is called as nucleoside. Okay, next. So thank you guys for watching my lecture. Thank you.
Thank you and thank you for watching.